All right. Welcome everyone to today's webinar, the first in our Beyond eDNA Basics series, focusing on the applications of terrestrial DNA. I'd like to start by acknowledging the traditional custodians of the country throughout Australia and their enduring connections to land, sea and community. And we pay our respects to elders past and present and extend that respect to all Aboriginal and Torres Strait Islander peoples today. As I look out of my window, I get to see the beautiful Adelaide rosellas playing in the trees outside. And for me, it's a daily reminder of how lucky I am to live and work on the country of the Kaurna people in the Adelaide Hills. Our lab and office are located on the lands of the Wurundjeri Woi-wurrung people in Brunswick, Melbourne. And so thank you to everyone for joining us today. There's been a lot of noise surrounding terrestrial DNA, and today we want to provide some clarity as to how your terrestrial surveys can give you the best chance of results. We think it will be approximately 60 minutes. We're going to stop halfway for a very quick Q&A session. You can also submit questions in the Q&A panel. A recording is going to be available, and any feedback or suggestions for future webinars are always gratefully received, and you can contact us at info@envirodna.com. And so why Beyond eDNA? So I was at the Biodiversity Offsets Conference last week in Canberra. It was very clear to me that people are very comfortable with using eDNA in an aquatic environment. But of course, we believe there's so much more that you can do with eDNA. And so through this webinar, we aim to really educate our customers and clients on what else is available beyond the very basics of eDNA to really provide greater impact to all of your projects. And so a quick introduction to who we are. We are a commercial eDNA lab, as I said, based in Brunswick, Melbourne. We were founded by researchers in 2016. We have an amazing team of field ecologists, molecular staff, data science experts. And just to give you kind of an idea of the impact that we have, in the last 12 months alone, we processed 40,000 samples, we detected 20,000 unique species, we detected 66 threatened species, and to date, we've developed more than 150 qPCR assays. One of the things that we really love to do is work with clients on very hard to find cryptic species and develop qPCR assays. So we'd love to chat to people about that. And so to introduce our speakers, first of all, we've got Josh Griffiths. When I asked Josh to send me his bio earlier in the week, he said, just call me the platypus guy. But he's actually so much more than that. Josh has been a wildlife ecologist for 30 years. He specializes in platypus, but he, as you will see today, has an immense passion for ecology that really shines through in the conversations he has with our clients and the support he provides, whether that's designing surveys or helping our clients to really understand the results and make sense of what all of this data means to them. He's also a really passionate communicator. He often spends his weekends at community events and education events, whether he's teaching people about ecology, the environment, eDNA, or of course his favourite platypus. We're equally as lucky to have Isaac Di Natale with us today. He's probably a new face to some of you who've joined our webinars previously. So Isaac was an aquatic ecologist specializing in avian assemblages and threatened species monitoring. He's got extensive experience in habitat restoration, including revegetation, biodiversity surveys, and ecological burning practices. He joined our team fairly recently as a new project manager, where he's delivering amazing service to our clients, delivering end-to-end project delivery spanning both fieldwork and our molecular genetic workflows. And just to highlight how awesome and amazing our staff are, Isaac is also an accomplished dancer. He's danced professionally for very prestigious dance companies around the world. He's lived in Europe, Australia and New Zealand, and danced at the Sydney Opera House for the Sydney Dance Company. So multi-talented people on our team. And yes, so quick walk through the agenda. Josh is going to talk through an introduction and give an overview of eDNA, talk about the challenges with terrestrial eDNA, give you guys some ideas of survey designs, sampling opportunities and metrics, and then we'll hand it over to Isaac, who's going to walk through quite a wide range of different case studies. And then at the end, we'll have time for Q&A. And so with that, I'm going to hand over to Josh.
Excellent. Thanks, Nat. Let me just get my screen up and running. Okay, so hopefully you can now see my screen. Yeah, so I'm going to give a very brief overview, some background information around what eDNA is. Some of you are going to be familiar with this, so if so, you can take a break for 5 minutes or jot down some questions in the Q&A to get ahead of the curve. But I know there's going to be some people that are quite new to this, so I don't want them to get left behind either. So at a very basic level, what is eDNA? Well, essentially it is genetic material that's shed into the environment by a whole range of different organisms. And our genetic techniques are now becoming sensitive enough that we can detect these tiny traces from all kinds of environmental samples. Whether they're freshwater or marine that many people are going to be familiar with, or some more sort of targeted terrestrial stuff that we're going to talk about today. And this can be a whole range of different organisms, from micro, bacteria and fungi through to blue whales and panda bears. So it's a really powerful technique. It's got a really broad distribution, which also makes it quite challenging to explain to people. So that's why we run these webinars, to kind of showcase people the different types of applications. One of the things that's important to recognise is that, of course, it's just one of the survey tools that we can use as wildlife managers or, catchment managers. And it's important that we understand that every survey technique that we have has strengths and biases. And it's really important to understand where eDNA can be used most effectively so that we're making the best use of our limited resources most of the time. And again, this is why we do these webinars, so that you can understand the technology and also showcase some real world examples so that you can see how it's brought to life. But of course, we're always here for advice. And this is what, particularly myself and Isaac like doing, is working with clients. So we have the eDNA expertise, you have the knowledge of your local area and species, and the combination of that gets us a better outcome for the projects. So we're always here. Don't hesitate to reach out with questions or ideas, even if they seem a little bit outlandish. Often those are the ones that generate the most discussion around our office. So they're the kind of things that we really like to be tackling. And so while eDNA in aquatic systems is pretty widely accepted, and many of you may have already had experience in that, there's lots of studies demonstrating how effective it is, how sensitive it can be, comparison with other techniques, etc. But the use of in terrestrial systems is less clear and less well developed. And that's really due to, there's some challenges in terrestrial systems that I'm going to talk about, but also there's a lot of other survey methodologies that can also be applied. So eDNA may not be the first choice in some of these areas. So today we're going to give you some examples of how and why eDNA has been used in these different types of environments for different purposes to help you decide whether it might be an option for your projects. So some of the advantages of eDNA, of course, as wildlife ecologists, we always want to minimise our disturbance to animals or the environment. So being a non-invasive technique is really important. We don't need to see, capture, disturb our species of interest. The sampling methodology can be really simple, and even in terrestrial systems where it can be fairly diverse, it can still be a fairly simple sampling technology, so you don't need specialised equipment and specialised training, which opens it up to things like citizen science projects, which has been a really, pleasing development that we've seen over the years. It also means because DNA can persist and travel in certain environments, you get greater temporal and spatial reach. So you don't have to be at the exact location at the exact time of your species of interest. We know it can be highly sensitive, particularly in those aquatic systems. It can be very cost efficient, particularly when we're doing things at large scale. And you've probably seen us talk about projects before at scales, like for my species, a platypus, we're doing things at scales that have never been dreamed of before, which is really exciting. Because of that high sensitivity, we can detect species earlier. So this is really important for biosecurity, invasive species, to try to control and eradicate them. And as I mentioned, there's a whole range of species that we're able to detect, from those microorganisms through to large vertebrates. So we can get a much better picture of the whole spectrum of biodiversity rather than just focusing on one or a small group of organisms. And I guess the one thing that's really important to take away from this little introduction is the two main types of sample analysis. And I'm going to try to make some really bad analogies here, so bear with me. So when we talk about eDNA analysis, there's really two different types of methodology that's used in the lab. And so first is our target species assessment. This is typically using a process called qPCR. And this is where we can detect a single species of interest. So we develop a genetic probe that targets a unique part of the genome of our target species. We test it against a range of other species to make sure that it's not cross-amplifying with them. So we know that it's only going to detect our species of interest, and it's a very quick and easy and simple process to do. And for those that are familiar with bird surveys, I liken this to a call playback survey. So if we play a call of a certain species, only that species will call back to us, and it works very effectively, but it only gives you data on that one species. On the other hand is what we call our biodiversity assessment, and this is through a process called metabarcoding. And if our target species is like a call playback survey, our biodiversity assessment is like putting out an acoustic monitor in a forest and just letting it record everything for a day or so, and then using, say, AI or an algorithm to detect all of our known species of interest from that recording. So we can target whole taxonomic groups. So we detect multiple species from the taxonomic group that we target, and they can be quite broad, like vertebrates or bacteria, or we can get a little bit more specific. So we can target in particularly sharks or decapods. And we can use a combination of different assays to improve that taxonomic range. So quick example of how the qPCR assay works. So we go out and we collect our sample in the field, our environmental sample, whether that's water or soil or air, whatever that might be. When that comes into the lab, it goes through a DNA extraction and purification process. And within that sample, we're going to have a range of DNA from all kinds of different organisms. We then apply our species-specific qPCR probe, which will only bind to its matching bit of DNA. And when it goes through that qPCR and amplification process, we then get positive or negative results for each of our samples, and we can map them spatially across our study area. So here we've got green is where we detected the species, red is where we didn't, and yellow are what we call possible detection. So they haven't quite hit the threshold that we require to be confident about detections, but it's potential that those species are there. Our metabarcoding analysis starts much the same. Collecting the samples remains the same. We still do our extraction process in much the same way. And again, there's a whole range of DNA from different organisms in there. And then we apply some fairly generic probes. So they target our taxonomic group of interest. So in this case, it was vertebrates. So it just pulls out the vertebrate DNA from that soup of DNA. It then goes through a sequencing process. So rather than just saying the probes bind or not, we actually get a short genetic sequence for our region of interest. That then gets compared to a reference library of known sequences. And so we can determine what the sequences from our samples belong to, and we can get a whole list of species from our samples there. Very powerful technique, gives us a whole heap of data. Slightly less sensitivity than qPCR, but the amount of data you get across the range of species, I guess that's the trade-off that we have when we're deciding about which analysis to use. Now, of course, when we compare at a very basic level, our bioinformatics is we get our genetic sequences from our samples, we match it to a library of known sequences, and most of the time that is fine. But of course, we don't have genetic sequences for every species in Australia or across the world, or with even every population within species. So we're dealing with an incomplete reference library. And what that can lead to is you get errors or misassignments of species, because your closest match might be your local species might be present, but if we don't have the reference sequence for it, the bioinformatic process is going to choose the closest match, which is likely to be a very closely related species, but it might occur in a completely different area. And so that just means we're not getting the best available data that we can. So what we do through our analysis is we take a step further from that bioinformatics and we go through a process that we call ecological validation. So that uses a range of other metrics that we can try to improve the confidence in that data. So looking at the genetic data, so how well our sequences match the ones that are in the library. How genetically distinct are our species of interest? So is it platypus, which is quite easy to distinguish from others, or is it a galaxid, which are very closely related to several others that are likely to be in the area? Do we have reference sequences for the other species that are in that area so that we know we can differentiate between those closely related species? And that's often a step that's really critical to understand your data, but often something that's missed here. And then finally, from an ecology point of view, do the results make sense? Are we picking up species where we expect them to be? So thinking about their ecology, their behavior, the kinds of habitats that we're going to find them. We can use external data sets like Atlas of Living Australia or GBIF to work out, are these species expected to occur there? And then we can also tap into I think it's well over 100,000 sequences now. We've got a massive amount of data, both at a species, but also a genetic sequence level to work out where has that sequence been detected before, what was it previously assigned to, and have we made any adjustments, some manual adjustments from that before. So there's a whole heap of metrics that we can tap into to essentially make sure that the data that we spit out in the end is a lot more accurate and you can have a lot more confidence in it. So let's move on to terrestrial DNA. So that's what we're here to talk about today. And essentially all the stuff I've just talked about in terms of the type of analysis remains the same across virtually every system or habitat that you're looking at. The real challenge for terrestrial systems is simply being able to locate the DNA in the environment. So it becomes a survey design and sampling issue. And unlike in aquatic systems where the DNA will diffuse and also disperse through the water column and with flow, in terrestrial systems, it largely remains static. So it means you have these traces of DNA that are very patchily located, and you can't simply do things like filter large volumes of water, which you can do in aquatic systems. So locating that DNA becomes a significant challenge to your survey or sampling design. The solution to that is we need to think outside the box a bit and come up with bespoke designs that are tailored to your environment, your species, and your objectives. And so unlike in aquatic systems where there's a lot of evidence about how effective eDNA can be, and it's often used as a standalone method due to its high sensitivity, in terrestrial systems, we're often implementing eDNA in conjunction with other survey techniques to provide that extra layer of monitoring and confidence. So things like wildlife cameras or acoustic monitoring in conjunction with our eDNA surveys. Excuse me, I'm already going croaky. And I guess the first step of this is we need to understand what eDNA is and how it can be used. And, that was sort of a bit of the intro there. And importantly, you don't need to be experts. That's what we're here for. But it is good to understand the basics so you can have an idea about when and how it can be applied. The important thing is, and even more important in terrestrial environments, is really thinking about your species of interest or your group of species of interest, their ecology, their behavior, their movements, how they might interact with other species. Are there key predators of your species of interest that we should be targeting? And using this information to target your sampling locations to areas of the highest activity so you can improve your chance of capturing that DNA. So targeting areas like common pathways or burrows or hollows, latrines or drinking holes. As I mentioned before, are there major predators that we can look at, their scats, to try to target your species of interest? And then we can get creative with sampling. So there's lots of different ways to collect DNA from these environments. And it's important to understand not all of these have been explicitly tested and validated. So often there's a pilot study step that needs to be required. But Isaac's going to run you through a whole range of different examples about how we can use different techniques in terrestrial environments. And so these are just some of the ideas. And so typically people, when we think about eDNA, everyone's very familiar with taking water samples. And of course, we can detect terrestrial species from water samples, but predominantly we're targeting those aquatic species there. But all these other techniques can be used as well. So scats are an incredibly great resource of DNA. And unfortunately in Australia, we have some amazing predators of our small mammals and birds. So they can be a really valuable resource to target if you're particularly interested in those kind of taxonomic groups. Soil, I'm going to touch on soil in a little while. Soil's not so much for targeting those larger animals, but thinking about microbiome of soil, soil health, and what that means for ecosystem health. We can use simple swab processes to target surfaces, or we can swab scats as well. One of the projects that I'm really interested in doing at some stage is using those predatory insects. So it's a process called something called eDNA. So using the blood that we get from things like mosquitoes or leeches or some of our predatory flies to look at what they're preying on. And of course, they sort of travel around and prey on a whole range of different areas. So if we can attract and capture those mosquitoes, we can see what's in those, particularly birds and mammals they're going to be preying on. Using swabs or even the petals themselves of flowers, so we can look at what's pollinating those types of species. Down the bottom here, we've got something that's called spray aggregation. So this can be either natural using rainfall, or we can actually artificially do it with hoses. So spraying down things like a tree, collecting that runoff and getting DNA from that broad area from a large tree to see everything that's kind of living in that ecosystem. Similar to swabs, we can use rollers to target surfaces, either leaves or tree trunks or branches. Again, thinking about where you're most likely to find your species of interest. And I think something that's really interesting in the terrestrial space is using EnviroDNA. So sounds like a bit of science fiction, but we can now, it's been proven and demonstrated that we can actually capture and analyze traces of DNA from air samples. And that gets around the main problem that we have in terrestrial systems of DNA not traveling very far. So it's more like those dynamic aquatic systems. And EnviroDNA, we've got a range of kits that can be used to target those different environments. So water, soil, scat kits. We can also work with you to design more bespoke area, sampling techniques. If you are looking at something that's beyond our sort of standard sampling protocols, we do ask you to get in touch so we can check with the lab around, can we actually extract DNA from those I guess, consumables easily and effectively. So that's something just to check with us. So, and I guess what's driven this webinar is that we get lots of inquiries about use of eDNA in terrestrial applications. So as a result, we've also been doing a lot of R&D in this space, some of which Isaac's going to touch on today. And I mentioned eDNA as being a really exciting issue at the moment. And so it's been demonstrated, there's numerous publications around it's possible to do. I guess what we're trying to understand now is a little bit about the nuances of it. So how can it be applied most effectively? What are the different sampling methods and protocols that can be used? What types of species or groups of species is it most useful for? And I guess conversely, which ones is it not very effective for? How do things like environmental conditions and habitat structures affect detection? So these are all sort of some of the, I guess, variables that we need to understand so we can apply eDNA a lot more effectively. One of the other questions that we get a lot is most people are trying to understand ecosystem health, and particularly how that might change either spatially or temporally in response to things like a natural disturbance or a management intervention. And we know that invertebrates can be a really good indicator group here. They're widely used in aquatic systems, and it makes sense that they're going to play the same role in terrestrial systems as well. So we've been trialing a variety of different things like a light trap that's mentioned here, and correlating that with different health metrics to see if we can use that as that broader ecosystem health indicator. So those are fairly broad scale things, but then we can get quite targeted, like swabbing the leaves of some agricultural crops to look at invasive pests, and can we pick them up earlier and more effectively so that we can implement controls. So there's a lot of work going on in this space, and we'll continue to direct our R&D in these areas that we see of most value for you out there in the various industries. Now, I mentioned soil health before. This is something that's been getting a lot of interest recently, and the role of that microbiome that's present in soils and how it's related to the health of terrestrial ecosystem. So this can be done in isolation or often in conjunction with other soil health metrics like a chemical or physical analysis. And it's really those microorganisms that are of interest here. So we're generally not looking for large kangaroos or wombats or anything. That DNA is going to be very patchy, and there's probably other techniques that are going to be more effective here. But in every gram of soil, there are thousands of bacteria and fungi. And now we have the methodology to be able to assess these quickly and effectively and use those as an indicator either for soil health or as that broader indicator of ecosystem health. And this is particularly powerful for activities like mine site rehabilitations, where you're starting from a very low base level and you can track how this improves over time, and using control areas, so nearby areas that are relatively natural and reflect what your site would have been originally. Use that as your reference site to see how your impact site is tracking against that. I did mention that we don't typically use soil to look for large organisms, but this is a study that did just that a few years ago. I think this came out in about 2017. They had camera traps out on some on the confluence of a couple of game trails, typical sort of wildlife monitoring using camera trapping. But they were really interested to see, well, could eDNA be effective in this space? So they then took soil samples in the viewfinder area of where the camera was looking, and then they overlapped what the detections were for the different methodologies. And so we ended up with this Venn diagram here where there was quite a lot of overlap. So we picked up these species with both techniques. There was one species that was only detected with the camera. And then there was a whole range of species, particularly small species that are hard to detect with cameras or hard to identify. So some of those rodents that it's a rodent, but you can't tell which one. So eDNA provided a really great complementary method here and actually extended the kind of species that could be detected. So this just highlights how it can be applied in terrestrial systems really effectively. And there's all kinds of metrics that you can derive from this, similar to what you're probably familiar with in an aquatic system. That's all relevant for terrestrial systems as well. We just need to think a little bit more around really the survey design and sampling protocols around how we can achieve this to the best effect. So I'm starting to lose my voice and I want to leave lots of time for Isaac. So we'll have a quick break now and go through a few questions, and then I'll hand over to Isaac.
Awesome. Thank you so much, Josh. Yes, so as Josh said, we have time for like 1 or 2 quick questions. If anyone has anything, please put it in the Q&A now. Tamika, I see you've got your hand up. Might need you to actually type it in if that's okay.
I've got one in the chat here, Nat. There's difference in cost between single species and biodiversity assessments. Yes, there is a cost difference there. The biodiversity assessments are more expensive, roughly around double the price of a single species assessment, but it does vary a lot depending on different assays that you're using, different sample numbers. But if you use that as a ballpark figure, around twice as much for those biodiversity assessments. I guess as I mentioned before, the trade-off is you get a heap of data on a range of different species. The trade-off is the sensitivity for some of those rarer species is a little bit less. So we need to think about what your goals and objectives are around which analysis is going to be best for you.
Awesome. Thanks, Josh. One more and then we'll move on. So the question is, for more bespoke species or targeted surveys that require pilot studies or lack published reference studies, how do you assess the effectiveness of eDNA sampling and the level of confidence in the presence or absence?
Yep. Isaac is going to give an example of this in a minute. But I guess, first of all, is having a good study design to start with. And then typically we would use something like a hierarchical site occupancy analysis to work out detection rates, incorporate things like false positives, false negatives, and also a power analysis to work out, well, what kind of survey effort is required to detect these species. Often it requires some sort of external data as well. So one, you want to know the species is present, and ideally some kind of idea of abundance, even if it's fairly generic, like low or high, so that if stuff's in high abundance, you're going to pick it up almost all the time. So you want to understand the variability across the sort of range of species occurrence.
Awesome. Thanks, Josh. All right, we will hand it over to Isaac now for the rest of the presentation.
Great, thanks. So I'll be running through a range of case studies to help highlight some of the ways we tackle terrestrial taxa. And whilst eDNA has traditionally been centered around water sampling, we've seen that when we pair that ecological context with a target species, and novel survey techniques, we can detect those terrestrial taxa which would otherwise elude some of those monitoring efforts. So when we consider a terrestrial target species, water eDNA can still be applicable. And that's because we can use what we know about a species' behavioral ecology to inform targeted survey approaches, particularly for species that are contacting the water frequently. So the broad-toothed rat pictured here is a small native rodent, and it's the only living extant species in the Mastochymus genus. So this is a species listed as nationally threatened, and they occur in low abundance. As well as being so small, they're not readily detectable with traditional monitoring methods. However, we know about this species that it has very specific habitat requirements, and they maintain quite local small home ranges where they do occur. So that's where we tie in that species behavioral ecology. They have a high fiber diet, and that leads to a high water demand. And we can then zone in on known or suspected drinking areas or suitable water sources within those limited home ranges to collect those eDNA samples. So in this project, we did just that, and we applied vertebrate metabarcoding targeting the broad-toothed rat. And using this approach, we were able to achieve a 10% detection rate from about 50 sites by leveraging that species behavioral ecology to provide some really valuable presence and distribution data for a species that's really persisting across multiple small isolated subpopulations. And because we applied that metabarcoding, we could have that supplementary data to also inform the ecological community that the species is occurring in. If I could go to the next slide, please. So as part of this broad-toothed rat project, we conducted eDNA power analysis to determine the number of sites and samples required for a reliable detection, conditional on the species truly being present at the time of sampling. So power analysis for eDNA surveys, we use species occurrence records to estimate the target species site occupancy and combine that with detection probability estimates from pilot data or previous studies or that generated with eDNA. So power analysis can also incorporate some variability into the model, your desired significance level, and the statistical power to determine the number of sites, water samples, or replicates that are required to produce a positive detection if that target species is truly present at the time of sampling. Because eDNA surveys are relatively rapid and cost-effective, they provide a really efficient pilot method to generate the detection and variability data needed to build out these robust power analysis models. And it also provides a scalable and repeatable method that you can use to accurately monitor species occupancy or population trends over those longer time periods. Next slide, please, Nat. So although a target species might be rare, unfortunately, as I'm sure many of us are aware, introduced predators are not. And in particular, those foxes and cats are really impactful predators, and they're prevalent in our landscapes, consuming a large number of small to medium native taxa. This does, however, provide us an opportunity to use them as aggregators of environmental DNA for rare and hard to detect species by using their scats to infer prey species presence despite that predation pressure. So in this application, we used fox scats to assess the presence of the long-nosed potoroo, which is a small threatened native marsupial, with fox and cat predation listed as primary drivers of the potoroo's population decline. So the scats were collected from the environment. In this case, it was by the client, but that can also be by trained detection dogs in many cases. When we had those fox scats in the lab, we applied qPCR to identify which scats belong to those common introduced predators and primarily targeting those foxes. And once a scat was confirmed to be from a fox, we then applied metabarcoding to identify the prey animal DNA within those scats. And we were able to confirm the presence of the long-nosed podroo within that survey range. So this method is applicable to detect a broad range of typically difficult to detect terrestrial taxa. So our small mammals, reptiles, and birds, anything that those cats and foxes are consuming routinely. And we do see rare and cryptic detections using this method quite often. Next slide, please, Nat. So we can also utilize the DNA within scat samples to help identify individual predators and assess the impact of predator control programs. And we do that with predator scat genotyping, using that eDNA recovered from fecal material from their scats to identify predator species through genetic analysis. So by genotyping multiple scat samples, we can start to distinguish individual animals and enable an estimate of population size, abundance, and distribution, including those camera-shy or trap-shy predators that would otherwise evade traditional control measures. So when you're getting down to the end of those control programs. So this non-invasive approach is particularly valuable for eradication and conservation, where confirming that number of individual predators that are remaining can help guide targeted management decisions and also help to verify program success. And the inverse is also applicable. So we can use this to determine population size and genetic health of prey species populations where scats have been collected. Next slide, please. We also see that scats are a great way to help identify visually conspicuous or cryptic species that would otherwise be difficult to tell apart in the environment. And as an example, many of our bat species, especially the microbats, are quite difficult to detect due to co-roosting with similar species. In this case study, we aim to detect the threatened Pilbara leaf-nosed bat by sampling soil within caves where they roost. So the sampling methodology involved the client collecting tubes of soil which contained a mixture of bat guano or their droppings from underneath those bat multi-species roosting sites, and they sent those cores directly to our labs. We then applied a qPCR single species analysis to help us determine which locations the target species, the Pilbara leaf-nosed bat, was occurring at, and we successfully mapped a number of those roosting sites for the species. We've also used this method to detect a whole range of bat species solely from scats or guano and soil collected from the base of suspected habitat trees or roosting areas. And additionally, we've used the insects that are found within microbat guano to infer which problematic pest species are affecting nearby agricultural crops on nearby farms, as well as to look at the beneficial impact that bats actually are having on pest control in the area. So scat analysis, it's highly versatile and dynamic, and it really offers a range of application beyond just single species analysis. And it can be a really highly effective tool for landscapes that lack reliable water sources to sample. Moving on, please. Thank you. So Josh touched on soil earlier, and yeah, soils host these complex ecosystems. They're made up of billions of organisms, and identifying that soil biome composition can be key to understanding some of those indicators of soil health and quality. So we routinely perform eDNA soil microbiome analysis to provide companies with thousands of those biological indicators, so metazoa, bacteria, fungi, as well as arthropods, to help assess soil health, ecosystem function, and in particular rehabilitation progress following mining or development activities. So by analyzing that DNA from within the soil samples, companies can help monitor biodiversity recovery and detect changes in that ecological condition over time, as well as independently verify some of those restoration outcomes alongside other conventional monitoring methods. So it's an approach that delivers very rich, comprehensive environmental data across a broad range of complex taxonomic groups that you would really struggle to monitor otherwise. So those really small microbial communities. And it reduces the need for intensive field surveys, as well as minimizing that disturbance to recovering ecosystems. And we see that soil biome analysis is increasingly becoming cemented as part of evidence-based rehabilitation, quite literally from the ground up, and it plays a key role in environmental impact assessments and some of those regulatory frameworks. Next slide, please. So we've also been involved in helping to strengthen biosecurity by using eDNA to enable early detection of priority pests and pathogens before some of those visible symptoms might appear. So we've developed and validated sampling approaches across some major avocado growing regions using eDNA to detect a broad range of fungi and arthropod pests. And specifically, we've developed targeted assays for differentiation of brown root rot and Phytophthora. And we've successfully been able to detect both of those diseases from soil, root samples, and wipes that were collected from the base of both healthy and unhealthy trees on those avocado farms. So this program generated almost 20,000 detections across 1,250 fungal and arthropod taxa. So that is an absolutely massive data set that we were able to generate here. And at the same time, developing and validating those targeted assays to identify some of those key avocado diseases. So eDNA-based surveillance provides the avocado industry in particular with a rapid and cost-effective tool to detect those emergent threats earlier. And it supports targeted management decisions to help protect future production, with the methods we've developed here also applicable across agriculture in general. Moving on. Thank you. So light traps, when paired with eDNA metabarcoding, we've seen a really effective method where insect biodiversity is the primary objective. So this case study was part of a broader initiative on Christmas Island to help assess the impacts of ant control. So the island has had an incursion of invasive yellow crazy ants, which have had really devastating flow-on impacts to that local biodiversity and the endemic species. And you might be familiar with those iconic Christmas Island red crabs that migrate onto land every year. So here the light traps were deployed in control and treatment plots that corresponded to before and after yellow crazy ant management intervention. And the light traps attracted and funneled a broad range of insects into preservative buffer, which is generally just ethanol. And that allows them to be preserved before being shipped and extracted and analyzed in the lab. So metabarcoding was applied to help estimate insect biodiversity and the changes in trends before and after those ant control measures. And I'm sure it's no surprise that that biodiversity showed a significant increase without the ants being present in the plots. Next slide, thanks, Nat. So we also aren't afraid to step outside the box of traditional monitoring approaches, and we routinely combine creative field methods with science-backed tools to help fill those knowledge gaps and uncover some of those insights that would have otherwise be missed. So recently, we've had a project to apply novel sampling techniques to help detect predators of the Capricorn yellow chat, which is a critically endangered subspecies of a small bird in Queensland. So predation pressure of both adults and eggs is remaining quite an ongoing struggle for the management of this species. So when the client came to us, we came up with creating these artificial nests, and they were then set up using a mixture of plasticine and quail eggs, painted to resemble the Capricorn yellow chat eggs. So these nests and eggs were then left in the field, and afterwards the eggs were swabbed with our EnviroDNA swab sampling kits, and the nests were rinsed with clean water and subsequently sampled using those EnviroDNA water sampling kits. When we amplified and analyzed the residual DNA left on those nests and eggs by predators interacting with them, we were able to detect a whole range of predator species across multiple taxonomic groups. So of course, things like foxes, dogs, and dingoes, but also a lot of birds of prey. In particular, the Brahminy and black kites, so some of those birds of prey, were consistent in every replicate, which shed some light as they were previously suspected to be predating the nest, but they weren't routinely being observed on camera traps, even though they were having such a huge pressure when we looked at the eDNA data. So there was also indication of some other incidental interactions from multiple other species in the area, and that included agricultural species such as cows, which pose a trampling risk to those nests. Next slide, thanks, Nat. So snakes, as I'm sure many of us might be aware, are a very difficult group of taxa to monitor, particularly when they're threatened and in low abundance. And they're also difficult to detect with eDNA. They have low shed rates, given they're covered with these hard scales, and they have limited active times and relatively slow metabolisms. But we have been having some success with terrestrial snake species and threatened terrestrial snake species by designing targeted surveys around species behavior and microhabitats. So firstly, some work on the broad-headed snake. So here the sample collection involved targeted swabbing of crevices in rock formations where the snakes would spend most of their time. In the lab, that was coupled with highly targeted qPCR assay development to help maximize amplification of that target species DNA and ensure those detections were accurate. There's also some ongoing potential with this project to expand sampling to include pooled water after rainfall at the bottom of those rock formations within their range, to assess if that water runoff is acting as an eDNA reservoir for the species. We've also successfully designed targeted survey approaches to help detect the threatened grey-headed snake. So this is also a terrestrial species, but they're known to bask in shallow pools of water within their range. So that provided opportunity to take water samples from those puddles or pools, and it was also coupled with in-house generation of a qPCR assay targeting the species, which helped facilitate successful detection. And an additional benefit in general to these targeted qPCR projects involving assay design and validation is that we also sequence related species to ensure detections of our target are accurate, as opposed to the DNA sequence recovered actually belonging to a closely related species. And as a result, we're helping to resolve some of those gaps in reference libraries in-house for quite a cryptic lineage of taxa. Moving on, please, Nat. So eDNA can also provide a really impactful snapshot for habitats that are difficult to survey with other traditional methods. So we've been involved in surveys to evaluate how eDNA can improve monitoring of threatened species that rely on difficult to access habitats, such as tree hollows, nest boxes, or burrows. So this is an approach that helps to overcome some of those limitations of traditional surveys by giving you quite a large data set or a snapshot of wildlife activity for those habitats without actually disturbing any animals that might be using them or requiring extensive equipment at every site. And we've seen that this has been quite impactful to map occupancy of species such as greater gliders, sugar gliders, and swift parrots in remnant forest patches, to help determine if hollow-bearing trees are a limiting factor on population growth and recovery, as well as which species are outcompeting to dominate that limited habitat resource. Next slide, thanks. So I'll spend a little bit of time on some air DNA, which is an area we're really excited about at the moment. So that airborne environmental DNA is really an emerging technique that enables biodiversity monitoring, particularly of terrestrial species at new scales. So this is a method that we know works. We've seen overseas zoo trials capture quite a range of terrestrial taxa. However, that's obviously within quite a confined range. So this is a tool our internal research and development teams are actively pursuing to optimize for our Australian ecosystems, to help generate comprehensive species inventories at scale, and then to provide a method we can use elsewhere. Next slide, please, Nat. Thank you. So we've been involved in undertaking some air eDNA monitoring within Mount Rothwell's predator-free reserves. And this is quite an ideal location for research, given the very natural environments. And we can couple that with our already existing understanding of species occurrence within those reserves. So our long-term field surveys trialed solar-powered active samplers and simple low-cost passive samplers. So the active samplers, they refer to a battery-powered unit that essentially vacuums that air, whereas the passive samplers are as simple as facilitating airflow through tubing or pipes, where it then passes through a membrane to concentrate the DNA. Across our current air DNA research and development, we've detected 326 species, and that's across animal, plant, fungal, and metazoan taxonomic groups, so the whole spectrum of biodiversity. And much of the work that focused on plantal and ant detections was conducted within Mount Rothwell, and that's been modeled against effects of meteorological factors to help refine our sampling and bioinformatic pipelines. Moving on, thanks. So this is another air eDNA case study within the Western Australian wheat belt. And this one was really impactful in demonstrating the potential of that airborne eDNA as a scalable approach, specifically for terrestrial biodiversity monitoring. So here, passive air samplers were deployed across 5 sites with some support from citizen scientists. And that allowed the DNA captured from the environment to be analyzed using vertebrate metabarcoding assays. So across samples, and this was only 5 sites that these were deployed, the survey detected 68 distinct vertebrate taxa with site-level biodiversity ranging from 22 to 34 taxa. So these metrics are on par with some standard water monitoring projects that we're performing every day. And that displays how much potential there is in the air eDNA space once optimized, particularly for landscapes that might be devoid of obvious water sources to sample. So think of targeted application along places like game trails, riparian zones, wildlife crossings, cave entrances, landscape features where wildlife tend to concentrate and move through. That's where DNA could be a really impactful targeted approach. Next slide, thanks, Nat. Thank you. So just a brief summary from my end. Terrestrial eDNA monitoring, it presents us with unique challenges because that DNA is not dispersing through terrestrial environments as readily as it might in water. But here at EnviroDNA, we have a range of techniques that can effectively detect terrestrial species and communities, with some of those creative approaches always rooted in best practice science. And to really drive home, it's understanding that ecology for your target species or community that's critical to helping design an effective and informative sampling strategy, particularly where we're trying to detect those cryptic, small terrestrial taxa. I think that's it for me. I'll hand back to you, Nat. Thanks.
Thank you. Thank you so much, Josh and Isaac. Brilliant presentation, and hopefully everyone found it fascinating and useful as I did. So before we just dive into the Q&A, a little announcement, I guess, from my side. So we've got a couple of exciting things coming soon. So the first is our EnviroDNA field app. So anyone who's been a client of ours in the past will know that actually providing the metadata can be a little bit clunky with the current format. And so we're in the final stages of our field app, which I hope will make the user experience much more user-friendly. And we are also working on advanced reporting of our EnviroIQ data intelligence platform. So there's going to be a whole lot of new metrics and data analyses that will be available to all of our clients. Again, not far off launching that. So yeah, coming soon. So bear with me. I can see that Josh has been answering a few questions as I've been moving the slides along. Okay, so Josh, and let me know if I ask a question that you've already answered. I'm just going to start at the top. How effective is passive versus active eDNA sampling for flying insect DNA?
So that trial that Isaac mentioned directly compared passive and active samplers. We didn't specifically pull out flying insect DNA, but in terms of overall diversity and particularly focused on mammals, birds, and plants, I think that project was. So active samplers will get more DNA at a per unit level. The advantage of passive samplers is that they're cheap, easy, you can deploy them quite widely. So when we had arrays of like 5 passive samplers with our active sampler, we were getting very comparable results there. And so a cost per effort is much lower for your passive effectively. Sorry, I don't know specifically about flying insects for that.
Yeah, we can try and dig out some data and share that after the webinar. Next question, what method would be used to extract eDNA from tree hollows or tree trunks? For example, potential squirrel glider habitat where we're not sure if it's squirrel, crofts or sugar gliders using camera trap photos. We've had a few discussions around this, haven't we, Josh?
Yes, we have. And I would say, so with your camera trap photos, and I use that as a bit of an analogy as well, like when you're trying to determine species from your camera trap photos, they're really hard to differentiate. It's a bit the same with genetics. Crefts and sugar glide have only recently been split. We probably can't differentiate between those two. We can with certain assays with squirrel and the sugar glider group, but they're very closely related. So using things like swabbing hollows or swabbing the trunks outside of hollows, thinking about their behavior and where they move along the tree to target those areas. You could also deploy things like, say, passive air samplers in trees that you know that they occur on. I reckon that'd be a really interesting approach. Or looking at things like if you've got owls in the area, looking at their predator scats from the owls to work out what they're preying on to potentially identify those as well. So there's a few different options there. And again, it's about understanding your habitat and your species behavior as to what's going to be the best approach.
Yeah, perfect. Thanks, Josh. Okay, you've answered this in the chat, Josh, but I'm going to ask it because it's a question we get asked a lot. So, do camera traps and other camera traps and other methods can be used to determine activity of animals and abundance as well as occupier presence? Can eDNA be also used for this, or is it only occupancy or presence?
Yes and no. So we can absolutely quantify the amount of DNA that is in a sample, and at a very coarse level, that abundance. Probably not very informative at a site or a sample level because there's lots of things that can influence that. But if you look at it across a variety of sites, then yes, that can give you an indication of abundance. But typically we would use a metric like site occupancy because that you can deploy eDNA techniques widely, easily. You can sample a heap of sites. You can look at how many, the percentage of sites that your species is present to indicate abundance. We do this with platypus. It works very well for those widespread disperse species. And we've actually correlated our occupancy analysis with our abundance metrics from live trapping, and they match up perfectly. It's just looking at things at a slightly different scale. So for occupancy, you tend to look at stuff at a much larger scale. When you're doing trapping, you'll indicate abundance, but it's typically quite localized. So again, I think depending on what your species is like, how wide it's distributed, this can be quite effective.
Awesome. Okay, bit of an unusual question this. Do you provide services for New South Wales clients? I can answer that absolutely yes. We have clients globally, Australia-wide, but to move on from that, Josh, if so, do you know if targeted eDNA method, soil or water collected for the detection of threatened species is accepted by the Q and specifically the Assurance and Biodiversity Stewardship branch?
That's a great question. And I see someone from ABS has actually responded, which is fantastic. And Beth would love to speak to you. Yes and no. So we've worked with various regulators over the time to try to see how we can get eDNA methods accepted and put into the recommendations. We get various responses we want to see it used more by people on the ground. So it's sort of a bottom-up approach. We have recently one of the snake species that Isaac presented before. We've actually got that methodology accepted as one of the approved methods. I think that was in New South Wales, Nat, was it?
I think so. I would have to double check. I can't remember. Sorry.
But yes, I mean, we would love to know what the best pathway is to be able to provide evidence for various species. We think it should be a bit of a no-brainer for aquatic species. Certainly for platypus, you know, the sensitivity to detect presence of platypus is orders of magnitude more than what it is if I go out and trap them. So, and there's heaps of evidence there that we can provide. For terrestrial species, it's a lot more bespoke and probably needs that but certainly it's starting to be applied. I did see some of the new regulations are coming out that had eDNA in for various species as well. So, but yep, Beth, we might reach out to you after this and have a long chat.
Yeah, and Josh, very quickly, can you just chat about frog species and how easy they are to find using eDNA?
Yes. Yep. So obviously being an aquatic species or semi-aquatic species for most of them, eDNA can be really effective. The one thing with frogs is that it's an incredibly seasonal detectability approach. So most frogs, when they're not breeding or in the tadpole phase, they're not aquatic. They're under a rock 100 meters away from the waterway. So sampling at the right time of the year is really critical for frogs, same as when you're doing call surveys. So again, it's a very clear example of where you need to understand that species biology. Also making sure you're sampling the right habitat. So we get people doing surveys in, flowing creeks for things like fish, and they also want to look at frogs, but the frogs are, 20 meters away in a little wetland that's off stream. And if they're not hydrologically connected, you're not getting that DNA dispersal. So can be really effective for frogs. Just need to be really cautious about time of year and habitat that you're looking at.
Awesome. I might get Isaac to answer this question. It's a great one. If an eDNA soil sample for a flying insect species is negative, but the plant swab sample is positive, does that indicate that the species is just landing on the plants, but not necessarily breeding in those sample soils? Tricky one.
Yeah, I guess it depends on where that insect is, and I suppose where you tie in what you know about its ecology. I mean, yeah, I guess it's probably obviously on the leaf if it's positive. So it's contacted the leaf, that's where we've got that DNA from. In terms of is it in the soil or not, soil sampling can be a little bit tricky, especially if it's something as small as an insect. If it's just landing on the soil, you're probably unlikely to recover enough DNA from something small to give you a positive hit, whereas it's probably spent a lot more time on that leaf at that stage, at the point at which you're detecting it. That's not to say you can't detect arthropods from soil samples, but I would also assume a flying insect is probably at a stage in its life cycle where it's not needing to be burrowing as much and spending as much time in the soil. So yeah, I guess it's context dependent depending on that species. If you know the species, I'd love to hear it so I can have a Google.
Perfect, thanks. Okay, for eDNA para analysis, you mentioned using species occurrence records to determine occupancy. What do you do if the location is data deficient? Josh, you want to answer that one, or Isaac?
Yeah, so the power analysis is actually done on the eDNA data that's generated. We're not looking at, I mean, we can do some very loose stuff using ALA records, but because there's not, generally for most species, there's not absence records, can't do any occupancy analysis. So we're doing that on typically pilot studies that we've designed specifically. And ideally, we do these pilot studies in several different areas where you've got, potentially high abundance and low abundance of your target species, so that then you can see the power analysis where you've got different densities of your target. So you need to actually generate the data first to do that power analysis.
Thank you, Josh. Okay, I think we'll make this our final question. So using rehab versus control site as an example, for which they have very similar data for traditional soil microbiome analysis, such as bacteria stress indicators, class totals, microbe diversity indicator, or fungi to bacteria ratios. Have we found that eDNA has been able to show differences where these don't?
Am I going to be stuck with this one? I can take that. I would say I'm yet to see anyone do a direct comparison with existing techniques and eDNA in this space. I would say it's a fairly new area and we're just dipping our toes into it. I'll also say that we are certainly not soil microbe specialists, but we can provide data for people to look at this. But one of the cool things we have done in the last probably 12, 18 months is looked at the microbe community in rehab sites of different ages. So using a space for time substitution, because we don't have 10 years of data that we can look at. And we see a direct correlation with age of the rehabilitation and those microbe communities. So some of those metrics that you mentioned, we haven't looked at specifically, but in terms of that general microbe community, and I think we looked at the fungal bacteria ratio as well, we certainly see a relationship with age of that rehabilitation and also getting closer to the reference site that we use in this instance as well.
Awesome. Thank you, Josh. Okay, we'll wrap it up there. Thank you again, Josh and Isaac, for all of your time and effort putting this together. Thank you to everyone who attended. I hope today that we really showed how creative we can get using different methods where, I guess, traditional monitoring techniques may fall short. So if you have, anything interesting, challenging, please reach out to us. We absolutely love these conversations. We love to get creative and figure out how we can help. As a reminder, you can reach us at info@envirodna.com. And yeah, we look forward to you joining us for the rest of the eDNA Basics webinar series. Thanks again.